Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia. It's my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science, artificial intelligence, deep learning, and statistical modeling across the diverse themes of biomedical and health sciences. Our selected theme for this year has focused on quantitative approaches in spatial multiomics for guiding of personal medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and the hurdles associated with advanced genomic, transcriptomic, and other omic methods, along with a wide variety of data analytic opportunities that are presented for data science, deep learning, AI, and spatial modeling. We are particularly excited for this year's Biomedical Data Science Innovation Lab program, where junior faculty members in the biomedical and computational sciences develop novel joint projects on the topic of quantitative approaches and applications for spatial multiomics using guided personalized medicine. Participants selected for our special program utilize the content from seminars like today's to as essential material for our culminating in-person manuscript and grant development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. For today's seminar, I am delighted to welcome our speaker, Hilary Kahler, who is from the University of California, Los Angeles, where she is a professor of molecular cell and developmental biology, where she studies the genetic molecular basis of quiescent and temporary state a temporary state during which cells do not proliferate, divide, or produce more cells. She earned her PhD in toxicology at Massachusetts Institute of Technology in 1998 and has held postdoctoral fellowships in functional genomics at the Whitehead Institute, as well as cell cycle control at the Fred Hutchinson Cancer Research Center in Seattle. And her efforts seek to advance treatments for diseases associated with excess cell proliferation, such as in cancer, or a lack of cell proliferation, such as in chronic wounds. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Dr. Kahler will lecture today on pan-cancer landscape of epigenetic factor expression predicting tumor outcome, where oncogenetic pathways that drive cancer progression reflect both genetic changes and epigenetic regulation. Her lab has found that tumors for 5 different cancer types separated into several different clusters based on epigenetic factors, and the clusters themselves predict outcome better than grade or epithelial to mesenchymal transition. As a pan-cancer machine learning model, they've deployed epifactor expression data for those 5 cancer types, successfully separating the patients into poor and better outcome groups. Her studies have provided an epigenetic map of cancer types and lay the foundation for discovering pan-cancer targetable epifactors. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I'll be synthesizing these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, thank you so very much, Hilary. We are really looking forward to your presentation.
Thank you so much, Jack. Thank you for the opportunity to tell you about some of the research going on in our laboratory. And thank you for that really generous introduction. I appreciate it. So I'm excited to be here to tell you, as Jack said, about pan-cancer landscape of epigenetic factor expression and how it can predict tumor outcome. So I want to start with a huge thank you to a senior member of my laboratory, Mithun Mitra, who's a project scientist in the laboratory and has been really initiating these projects, managing these projects, leading these projects. And I wanted to start with a huge thank you to him. It's really been a pleasure to have the opportunity to work with him. So I'm going to tell you about a couple of projects, all of which culminated in the project that Jack was talking about. So you get a better sense of how we got to where we got to in terms of trying to understand intertumor heterogeneity. So all of this really started when Dr. Mitra came to me and he said, I really want to understand intertumor heterogeneity, meaning why is it that people who have the same type of cancer have such different outcomes? So people can have tumor of a specific site. Some of them will progress rapidly and some will survive for a long time. And he said, I really now, like here in the work that we were doing leading up to our paper in 2018, he said there's a lot more data available so that we can start asking questions about this issue of intertumor heterogeneity. And so we started by looking at triple negative breast cancer, and we chose triple negative breast cancer because it's a tumor type that is relatively common. Many women die of it. It is particularly aggressive, and we really don't have good therapeutic strategies. And so we wanted to try to get an understanding of why is it that some patients with triple negative breast cancer will die so quickly, and how come that some will survive for longer. So triple negative breast cancer is characterized by a lack of the estrogen and progesterone receptors and HER2 amplification. And as a result, most of the approaches that we have for treating breast cancer don't work for these patients. They're known to be heterogeneous in terms of genetic variations, histology, and clinical outcomes. And so in this paper, we used high-throughput genomic data from triple negative breast cancer patients, so 137 of them, from TCGA, which has been a fantastic resource for us in order to get a better understanding of intertumor heterogeneity. And as I'm going to show you, we use something called a similarity network fusion-based integrative clustering, and that's clustering that allows you to try to take different types of data to try to get a better understanding to underlying mechanisms for triple negative breast cancer. So this just shows you from previous publication just how heterogeneous triple negative breast cancer is from a histological perspective. And so all of these different images show examples of triple negative breast cancer. You can see from the perspective of pathologists, it really is quite heterogeneous. And there had been some previous studies that had tried to classify triple negative breast cancers based on gene expression patterns. So Chuck Peru has the PAM50 that had been applied. And there are other approaches as well to try to understand breast cancer and even specifically triple negative breast cancer in order to get a better understanding of what are the subtypes of triple negative breast cancer with the hope of being able to see are there patients with certain types that would benefit from specific treatments. And so the goal of our work was to better define the molecular nature of this intertumor heterogeneity in triple negative breast cancer patients in terms of gene expression, microRNA expression, and copy number variants. So we were really excited to have access to through this Cancer Atlas, these different types of data. And our hope was that maybe by integrating different types of genomic data, we'd be able to do a better job of understanding the underlying substructures of triple negative breast cancers, and maybe even to identify novel genes that could be targets for specific new subtypes. And so the approach that we used was this similarity network fusion. So we took RNA sequencing data, microRNA data, and then eventually we would use copy number variants as well. And we turned these into count matrices. And then we used this similarity network fusion, this SNF approach, in order to try to combine these different types of networks. And the program, the idea was that it was going to be able to weigh the different types of information and maybe give a little bit extra emphasis to information that's particularly informative. And we found that if we used gene expression only to try to form clusters of these different patients, we could form clusters, but they weren't particularly well differentiated from each other. If we use microRNA data only, we could form clusters and we could kind of see the data separating a little bit. And this group out here seemed to be kind of separating from the others, the group in blue. But again, they weren't very distinct. And then we found that if we combined the gene expression data and the microRNA data, then this group in blue was really becoming much more distinct. So this was really exciting to us that somehow the gene expression data and the microRNA data were providing kind of orthogonal but complementary information about the fact that there was this subgroup of triple negative breast cancer patients that seemed to be somewhat different from the others. And then in the next phase, we combined the gene expression data, the microRNA data, and copy number variation data. And this was actually particularly exciting to me because when we combined all three different types of data, we could see that this cluster 2, we called it, group of patients, was really separating even further. And so this suggested to us that there really is a very different group of triple-negative breast cancer patients. Tumors have different characteristics. We can see it somewhat by gene expression. We can see it somewhat by microRNAs. We can see it somewhat by copy number variants. But when we put all of that data together, that group really pops out. And we looked at the cluster 2 versus cluster 1 and cluster 3 based on previous analyses of different types of breast cancer. And we could see that maybe there were some differences in these previous compared with these previous breast cancer kind of categorizations. But there was no single subtype that had previously been characterized that cluster 2 was in. So the type of kind of clustering that we developed seemed to be complementary to and not exactly the same as anything that had been discovered previously based only on gene expression. And we looked to see whether patients in cluster 2 had different outcome. And we found that actually, with a very strong p-value, patients in cluster 2 actually had a worse prognosis. So if you look at the percentage that are disease-free, it decreases much more rapidly for patients in cluster 2 than for the entire data set. And we didn't see that for patients in cluster 1 or cluster 3. And when we looked, we could see that patients in cluster 2 were a little bit more likely to have a later stage, so to have a worse prognosis, and to have a higher pathological T, which is a sense for whether the tumor has spread to lymph nodes and its size. So in conclusion, we found that these similarity network fusion-based integrative clustering, combining gene expression, microRNA expression, and copy number variation, revealed 3 distinct patient clusters, and integrating multiple types of data resulted in more distinct clusters than analyses with a single data type. So whereas most triple-negative breast cancers are classified with the PAM50, the gene expression, as basal cell type, one of the clusters that we found was enriched in kind of non-basal PAM50 subtype and had more aggressive clinical features. We think that this may reflect the way in which these cells got to the triple negative state, and we think more research would be exciting to try to follow up on this. We also noticed that the androgen receptor was higher in these patients, and so we thought that this might suggest anti-androgen therapy for these patients, which has been being tried in triple negative breast cancer. Overall, our analysis provided a new classification scheme for triple-negative breast cancer based on the integration of multiple omics data sets. So then we moved on to analyzing another really aggressive and terrible form of cancer, which is pancreatic ductal adenocarcinoma, or PDAC. And for these studies, Dr. Mitra approached me and he said, I really want to study PDAC because it's a super aggressive cancer with very poor prognosis. And in this case, we realized that now, given all of the RNA-seq data that was becoming available, it was possible to look not just at the levels of different genes, but also to look at splicing. And so the idea here was to ask, is alternative splicing or some form of alternative splicing actually an important contributor to tumor outcome, to intertumor heterogeneity? And we were going to take a look at that in the context of pancreatic ductal adenocarcinoma. So just for background, PDAC, or pancreatic ductal adenocarcinoma, is an aggressive cancer with a 5-year survival rate of less than 8%. There are 4 genes that are frequently mutated in PDAC, so KRAS, P53, SMAD4, and CDKN2A. But nevertheless, these genes by themselves and their mutations can't really explain all of the intertumor heterogeneity that's observed. And there's a lot of heterogeneity at the genomic, metabolomic, transcriptomic, and histopathological levels. And that's shown a little bit more here. So PDACs can have different types of structural rearrangements of the chromatin. There's a lot of heterogeneity based on histology. And again, just like the triple-negative breast cancer, these tumors can look wildly different to pathologists. And there's heterogeneity based on gene expression. And some previous studies had attempted to subclassify PDACs based on gene expression patterns. So the goals of this project were to better define the molecular nature of intertumoral heterogeneity in PDAC patients based in terms on variations in alternative splicing. We're hoping to identify molecular subtypes of PDAC through alternative splicing data and correlate them with clinical outcome. We're hoping maybe we'd identify novel splicing targets and differentially spliced events, and maybe even to identify RNA binding proteins that could be responsible for any differential splicing that we found. So we had a pipeline for analyzing these PDAC patients, getting their data from TCGA, and using different methods that allowed us to develop clustering of the patients based on different types of splicing. And here we're using non-negative matrix factorization, which is a clustering technique that's going to decompose the data into lower dimensional matrices. And that allows us to try to find underlying patterns and structures in the data that can be used for clustering. So we used 5 different types of alternative splicing to develop clusters. And these heat maps that you see here are showing the clusters that we found for these different types. And this includes, we looked at exon skipping, multiple exon skipping, so when exons just aren't included or many times, alternative 5' splice sites, so at the beginning of a splice site if you use the canonical one or a non-canonical one, and alternative 3' splices, so at the end of a splice site, do you stop at the canonical place or do you use a different one? And when we tried to form clusters with these, we could form clusters, but these clusters were largely kind of symmetrical, meaning there were some tumors that for some gene, they tended to exon skip. For other genes, they didn't exon skip. A different tumor would be more likely to exon skip in some place, less likely to exon skip in a different place. And so we could form clusters. But intron retention really stood out. And for intron retention, we saw that there were certainly times when an intron was retained, meaning that something that is normally spliced out was still there in the final transcript, and times when it was removed. But we found that some tumors were really big splicers, so they rarely retained introns, and some tumors usually retained introns, so they were kind of low frequency splicers. And this was true over, if a tumor was a splicer, it tended to like almost always splice and almost never retain an intron. And if it was not a splicer, it tended to like retain all the introns that we were looking at. So we considered this an asymmetrical division among the clusters. And we specifically saw it for intron retention. And when we looked at the 2 clusters that we generated based on intron retention and compared them with the previous classifications for PDAC, and so those are shown here, we saw that the clusters that we had made didn't align with any of the previous classifications. And so this just shows, again, what the PSI, so the percent spliced in. And we can see that for these different types of splicing events, most of the events were symmetrical. So some tumors did more splicing at some places, less splicing at others. But for intron retention, it was very asymmetrical, meaning a tumor either always did it or never did it. So it seemed to be a kind of a quality of the tumor, a characteristic of the tumor, that it would retain these introns or not, all throughout the genome. And again, this is showing you that the clusters that we identified for intron retention 1 and intron retention 2, so these intron retention clusters, had very little to do. There was really no correspondence with these previous examples of ways to characterize PDAC. And this again shows a couple of different ways that you can classify PDACs, and again shows that intron retention 1 and intron retention 2, the 2 clusters that we found, didn't seem to overlap with them. And overall, we didn't seem to see much association with these clusters in terms of sex or age or these different pathological features, such as pathological T or N, stage or grade. Yet, even though we didn't see correlation with any of these other features, we actually did see a big difference in progression-free interval, disease-specific survival, and overall survival. And we found that intron retention cluster 1 tended to have a lower progression-free interval. It had worse disease-specific survival, and it had worse overall survival. And so these was actually very interesting for us, because even though it didn't classify and didn't correlate with stage or grade, we're actually seeing a difference in clinical outcome. And if we compared not just to intron retention 2, if we compared to the entire cohort, we saw that the tumors in cluster intron retention 1 had a worse survival. And also, we tried to take an independent data set. So this is another data set that's completely independent. We use these same features, intron retention 1 or 2, to analyze that data set. Again, we saw a lot of asymmetry in that most of these sites were either had retained introns for all the sites or almost none of the sites in this independent data set. And again, in this independent data set, we found that the tumors that were similar to intron retention 1, had worse survival for progression-free interval, disease-specific survival, and overall survival. So when we looked at percent spliced in, so how often you're doing splicing, we saw a big difference between intron retention 1 and intron retention 2 groups. And you can see that classified, that's shown here, where intron retention 1 tended to have less splicing, intron retention 2 more. And you can see that here. So with intron retention 1, you can see that there was almost always spliced. So almost never did we see these introns being retained for intron retention 1, whereas intron retention 2, they tended to have hold on to their introns. So intron retention 1 are the really good splicers. They tended to have worse survival. Intron retention 2, they tend to hold on to their introns, and they had better survival. And so we wanted to dig into this further and see if we could understand something about the possible mechanism. We looked a lot at the genes that were being differentially have intron retention. And one thing that really stood out to us was that oncogenes were among these genes that were more likely to be some of these genes that where the introns were either retained or not. And so this was very interesting to us because we know oncogenes are important for promoting tumor progression. And we could see that these genes that in the intron retention 1 were tending to be spliced, and the intron retention 2 weren't tending to be spliced, were enriched for oncogenes. And when we looked at gene ontology categories for these genes that sometimes were being spliced and sometimes weren't being spliced, here too, we saw that there was an enrichment. In this case, there was an enrichment for genes that were actually involved in RNA processing and splicing itself. So this suggested to us that maybe there's some sort of feed-forward loop where these proteins that are important for splicing are themselves being spliced or not spliced, and this will make them more or less active, make them more or less available to do splicing on other proteins. And so we looked more carefully. So there are 259 RNA binding proteins that we found were differentially expressed between intron retention group 1 and intron retention group 2. And so here you see the gene expression heat map for these 259 differentially expressed RNA binding protein genes between these two clusters. And we also looked for motifs, so RNA binding protein motifs that they recognize. And we found the logos, so the sequence recognized by these RNA binding motifs. And we found 30 of them that were enriched in these retained introns. And some of the important RNA binding proteins that we found their logos are shown here. And we found that the expression levels for 25 of these RNA binding proteins actually correlated highly with the extent to which they were undergoing intron retention or not. So these 10 RBPs with the highest number of highly correlated intron retention events are shown here. And also importantly, when we made protein-protein interaction networks, we could see that these proteins that themselves are spliced, that themselves their expression levels are different between these patients in these two different clusters, form protein interaction networks. And so this furthered our hypothesis that perhaps what's going on here is not a mutational mechanism necessarily, but the lack of splicing can lead to more lack of splicing or more kind of processes involving the RNA itself, even without genetic mutations. So that's one of the things that I think was so striking here, was this suggested to us that even without mutations, a cell could go in one direction or another, having more and more of these RNA binding proteins, either that are doing a good job splicing other RNA binding proteins and doing more splicing, or it could get into a mode where its splicing is not good. The proteins that are supposed to do the splicing are not as effective because they're getting their introns retained. And as a result, it gets into kind of a bad splicing mode and ends up having more retained introns. So with this hypothesis in mind, we went and we looked at other tumor types, and we could see when we looked at a bunch of other tumor types, we often saw asymmetry in intron retention in those other tumor types as well, showing that this process didn't seem to be specific just for pancreatic ductal adenocarcinoma. And in the case of two other tumor types, so Kirk and Pratt, we saw not just asymmetrical intron retention patterns and two clusters, but here we also saw that there were differences in the survival outcome. So here's the survival plots for Kirk and Pratt, and here we used our intron retention events that we identified in PDAC and saw that when we made two clusters, they had differences in their outcome as well, showing that this like pattern of increasing or decreasing intron retention could predict intertumor heterogeneity and survival, not just in PDAC, but in other tumors. And when we looked, we could see that there were actually 194 common RNA binding proteins between the three different types of cancer, the PDAC, the PRAD, and the Kirk. And that showed us that perhaps there's a commonality, that there's a common set of RNA binding proteins that tend to be regulated in this process that can then go and regulate intron retention in other proteins. So taking all of the data that we put together for this project, we came up with kind of a working hypothesis or a working mental model of how you could get differential outcome for these two different types of cancer, all PDAC, but the intron retention 1 cluster and the intron retention 2 cluster. The model that we have in our minds is that for the tumors in intron retention 1, they're doing a good job removing their introns. They're making RNA binding proteins that are properly spliced. Those RNA binding proteins that are properly spliced are getting exported. They're getting synthesized. They're coming back. They're making functional RNA binding proteins that then go and do a good job removing introns on other proteins, including, importantly, oncoproteins. And as a result, you have properly spliced, exported, and translated oncoproteins that can then go and lead to worse outcome for the patient. In contrast, for the patients in intron retention 2 classification, here we see that there's more intron retention. This increased intron retention probably reflects the fact that these RNA binding proteins themselves aren't as effective. There aren't as many of them. We think that they're actually getting detained in the nucleus because we see that we see less of the properly spliced form, but we see more of the total transcript. So we think that these RNA binding proteins with the intron being included in them, are being detained in the nucleus. As a result, there's less of them being properly exported, there's less of them being synthesized, there's less RNA binding proteins available to go back and do proper splicing on other messenger RNAs, including oncogenic pre-messenger RNAs. And then those pre-messenger RNAs are including introns, they're getting detained too. And so you end up with actually higher levels of the transcripts, which we could see, but lower levels of the properly spliced form that gets exported and lower levels of the final protein. And so our thought was that perhaps in the tumors in this intron retention 2 cluster, part of the reason that the patients actually do better is because they have lower levels of these tumor-promoting oncogenes. So just to conclude, we found that intron retention produces the most statistically significant and robust clustering out of the 5 that we looked at. The intron retention helped to explain an independent dimension of heterogeneity in PDAC compared with other studies that have been done before based on gene expression levels. We found that intron retention is the only asymmetric alternative splicing event, and it occurred at lower levels in low clinical outcome patients. A significant portion of genes undergoing differential intron retention are themselves involved in splicing, which is very interesting to us. We found that retention of introns in oncogenes was significantly upregulated in the poor clinical outcome patients. We found that splicing differences between the 2 intron retention clusters can potentially be explained by differentially spliced splicing factors and RNA binding proteins. And also we found that intron retention was important for 2 other different types of cancer as well, suggesting that our findings could be more generalizable. So now I'll tell you about our most recent publication, and project on pan-cancer landscape of epigenetic factor expression predicting tumor outcome. So just as background, we started this project by pointing out that cancer has been associated with genetic disease with driver mutations in oncogenes and tumor suppressors. The project that we just did suggests that there might be non-mutagenic drivers of cancer as well. In this project, we wanted to had kind of two concepts going into it. One was to look pan-cancer. So we had been looking at triple-negative breast cancer, and we looked very specifically at pancreatic cancer, but then found our findings were more generalizable. In this case, we started with a pan-cancer analysis, trying to see what's common among many different cancer types. And in this case, we also came in with the question, okay, we know that there are epigenetic factors. We know that there are things like DNA methylation, histone modifications, other proteins are involved, for instance, in organizing chromatin, placing nucleosomes in particular positions. There's more and more evidence that these epigenetic factors are actually very important for tumor progression, even in addition to mutations and, so, for instance, copy number variants or chromosomal aberrations that affect the actual sequence of the DNA. And we wanted to understand, are these epigenetic reprogramming events, could they actually be important for tumor progression and for intertumor heterogeneity as well? So this was the question that we wanted to ask going into these experiments was, are these changes that don't have to do with changes in the sequence of the DNA or chromosome breaks, but could these changes actually be important for tumor progression? And as we did these experiments and as I read more, I became more and more aware of cases in which there was a tumor, but now we can sequence the entire tumor, and actually the tumor didn't have any mutations or any copy number variants that could help to explain it. So we were very interested in asking, is it possible that these epigenetic modifications are actually really important drivers of patient outcome and contribute to intertumor heterogeneity, which is the topic that we've been interested in. So the goals for this study were to investigate epigenetic heterogeneity across high purity, and so we were very careful to look specifically at high purity samples, primary TCGA, so from the Cancer Genome Atlas patients from 24 different adult cancer types based on the expression of epifactors. So in this case, we're going to say we're going to look at the gene expression, but only of a subset of all the genes in the genome, and we're going to ask, is that a predictor of how patients will do? So take away anything mutational, take away the entire genome, just look at the expression level of these epifactors. And for each tumor type, we're going to ask, can we cluster the tumors based on the expression of these epifactors? Does that relate to clinical outcome? Which epifactor signatures are important for defining these clusters, and are they common across cancer types? How does the epifactor landscape of adult tumors compare with pediatric tumors? And what's the relationship between inter-tumor heterogeneity and intra-tumor heterogeneity, so kind of cell-by-cell heterogeneity? So this shows you the 24 different adult cancer types in the TCGA repository that we wanted to look at. And so you can see all throughout the body, men and women, just many different cancer types, all different tissues of origin, all different cells of origin. And our idea was to try to look across different cancer types, are there commonalities? And we also used a previously identified set of 720 epifactors that were identified in an epifactor database. And these epifactors had a wide range of functions. These include histone modifiers that read, write, and erase histone modifications, proteins involved with histone folding and assembly, chromatin modifiers that change the formation of chromatin, proteins that act upon DNA and RNA modifications, so for instance, DNA methylation, and cofactors of these epigenetic complexes. So there were 720 of these epifactors in total, and it's important to note that only 164 of the 720 had ever been identified in the cancer databases as being mutated or deleted or overexpressed. So the vast majority of these epifactors, no one had ever seen that they were themselves ever mutated in cancer. And again, we use non-negative matrix factorization in order to take our big data sets, simplify them, and then look for kind of patterns in the data that will allow us to assign the patients to different clusters. And we looked at all 24 different TCGA types. And when we looked at the clusters that we got with this epifactor expression, so only looking at the expression of epifactors, we asked what's the best number of clusters to form. And for each of the tumor types, we looked at 2, 3, and 4, and we used 4 different metrics for how robust the clusters were based on how well separated they were and their cohesiveness. And for all 4 different tumor types, what we found was actually 2 was the best. So independently, we found that 2 was the best. And here's an example of BRCA, so breast invasive carcinoma. And you can see this heat map where we have a cluster 1 and a cluster 2. And there were genes that tended to be expressed high and genes that were expressed to be low in cluster 1 versus cluster 2. And so these genes were kind of informative. And here you can see for 5 different cancer types, the clusters that were formed by this epifactor expression. And here's some principal component analysis plots. So we took the different tumors and separated them based on their principal components. And you can see that the clusters that were formed by the NMF method led to 2 kind of clumps of tumors that could be kind of separated based on these principal component analysis plots, which are kind of low dimensionality projections of all of the data. And here you see for 5 different tumor types, we had 2 different types of 2 different clusters, and you can see how they project. And another thing that we did was to take all of this epifactor expression data and generate a map from all of the different cancer types to try to see how do these different tumor types relate to each other if we only look at the level of expression of these epifactors. And actually, we found that if you take all of the tumors and put them onto a map like this, we capture actually a lot of the information that you would get from looking at the entire genome. So looking at just 720 genes, we could recreate a lot of the relationships in terms of certainly all the tumors of the same type are close to each other. But for instance, you can see here 2 different types of lung cancer are physically close to each other on this map. 2 different types of brain cancer here are physically close to each other on this map. 2 different types of kidney cancer. So just the expression level of these epifactors was actually sufficient to recapitulate a lot of the information that you would get from looking at the entire genome. So this kind of tells us that the expression levels of these epifactors is really tissue specific and informative about the different types of tumors. And for each of these different types of cancer, we could ask how many top NMF genes there were. So the NMF clustering algorithm not only gives us the cluster, but also feeds us kind of the top most important genes for determining those clusters. And for the different types of tumors, we could see different numbers of genes were important and informative. And then we could look at the overlap among the different types of cancer at these NMF genes. And you can see that there were some kind of clusters here, and you can see some here, where there tended to be a lot of overlap among the informative NMF like identified genes for these different types of cancers, comparing them to each other. So that gave us some information that for certain types of cancers, the dividing the tumors into different clusters was being driven by similar genes. And among these, we took a look at what they were. One finding that we had at this point was that SATB1 was frequently one of the top NMF genes among the different types of tumors. And SATB1 is a chromatin organizer, so it helps to decide where nucleosomes go. So we thought that that was a very interesting finding. So then we wanted to ask, okay, these clusters that we made from the NMF based on only epifactor expression, do they result in changes in clinical outcome? And we could see that it wasn't true for all 24 types, but we saw, especially for this group of 5 tumors, we could see that when we looked at progression-free interval, disease-specific survival, and overall survival, these 2 groups did have a really significant difference. And that was true for these 5 types that, for me, before we did the experiment, I would not have been able to guess that these 5 types would be the ones that stood out. And so this just gives you, this was the most striking result for adrenocorticocarcinoma. We saw a really striking difference in the survival for patients in these 2 different groups based on the epifactor expression. So this is for progression-free interval. And you can see here for the 4 other types of tumor that are in this category of these, like this 5 cancer group, you can see again statistically significant difference in progression-free interval for patients in one cluster versus the other that we identified from the expression level of these epifactors. And when we looked at disease-free survival, again, we saw a statistically significant difference for these 5 different types of tumor in the 2 clusters generated from the NMF algorithm based only on epifactor expression. So then we did some controls. So we controlled for stage and grade. We controlled for the purity of the tumors and the amount of immune infiltration. And most of the epifactor clusters were still significantly different even after we did this correction. And as was pointed out in the introduction, so thank you, Jack, it was pretty surprising to us that for these 5 tumors here at the bottom, the predictive power of the outcome for the patient that we could get just from the expression level of these 720 epifactors was actually stronger than if you looked at their grade, which is supposed to be a really strong predictor of patient outcome, or something that we discovered was actually a pretty strong predictor of outcome, which is the extent of epithelial to mesenchymal transition. So that's how many epithelial cells had made the transition to being more mesenchymal-like, which we think is associated with kind of invasion and metastasis. So for at least for these 5 tumors, the NMF clustering based on epifactor expression was very strong. Which was very surprising, actually, to us, because grade was supposed to be really just the gold standard for whether a tumor is going to progress or not. And we compared the epifactor expression clusters that we found with existing kind of characterizations of these tumors based mostly on gene expression or methylation patterns. And we saw that none of the clusters were composed of only one kind of subtype, but sometimes there was enrichment. And this is methylation pattern, and this is a good example. We also wanted to take a look to see whether the epifactor expression patterns were correlated with the immune system. So the immune system we know is very important for attacking tumors, eliminating tumors, and closely correlated with patient outcome. And we could see that in some cases, the epifactor expression kind of cluster that we identified. So those tumors were associated with a more severe immunological phenotype if the patient had a worse outcome. And so these immunological phenotypes had been previously identified, and we could see some shifts. And in each case, if we saw a shift, we could see that going to kind of a worse outcome was associated with a sort of shift towards kind of a worse immunological phenotype. So that seemed to be correlated with what we were observing. We wanted to ask again, are these changes in epifactor expression that we're seeing, could they be explained by any existing mutations or copy number variants? So there's certainly mutations in these tumors. There's certainly copy number variants in them. When we looked, we tried to see, are there consistent mutations or consistent copy number variants that could help to explain why the tumors in this category versus the tumors in that category had very different survival outcomes. And what we saw was actually I had a very hard time finding any specific mutational events or copy number aberrations that could explain the difference between the tumors. Once in a while, they would have the mutation in this particular gene, but usually there were like many, many other tumors in that cluster that also had good or bad outcome that didn't have a mutation in that gene. The only one that looked like it could potentially have a role was P53. In the case of LUAD, we could see that P53, the fraction of the tumors with a P53 mutation was different in the 2 different clusters. But in the end, when we looked at whether P53 mutations could actually be responsible for it, we actually controlled for P53 mutations, and we still saw a difference in outcome. So it seemed unlikely that P53 mutations by themselves could explain the differences that we were seeing, or really any mutational event. So then we took a look at the top gene ontology terms when we took the gene expression changes that we were looking at with these epifactor clusters. We associated them with went back to all the genes in the genome and asked with whole genome network analysis, these gene expression changes that we're seeing, what do we capture that looks like them if we can take a look at the entire genome? And when we did that and took a look at the entire genome and asked what was correlating with our gene expression clusters, we saw, as you can see here, an enrichment in the dark orange module. That's one of the modules that come out from this like whole genome analysis for cell cycle. And we saw that for ACC and LIHC. So it seemed as though perhaps some of these epifactor expression level changes and some of these epifactors are associated with the cell cycle. We're capturing some information about the cell cycle. And again, we made kind of protein-protein interaction networks, and we could see protein-protein interaction networks among these epifactors that were being expressed. And in some cases, we saw kind of clumps that seemed to be cell cycle associated. We also thought that it would be really important to take a look at whether the changes that we were seeing in these epifactor expression levels actually correlate with an epigenetic change. The best that characterized one, and the one for which we could get the most information, was methylation patterns. And so adrenocorticocarcinoma has been previously classified based on methylation patterns. And we could see that our poor and better clusters that we generated, that's in pink and blue, did correlate with the previous identifications of extent of methylation, and that's shown by the shapes. So we can see kind of more circles up here and more squares down here. So we could see that there was some overlap between the methylation patterns and our epifactor expression, which makes sense because one of the things that we expected to capture with epifactor expression was going to be how much methylation there was. But we also point out that the separation that we get between the clusters when we look at all of the epifactors seems to be better than the separation you get just looking at methylation patterns. And that was true for other tumors as well. So when we looked at the fraction of NMF genes that were individually prognostic for the different tumor outcomes, we could see that there were quite a few of these like NMF genes were actually individually prognostic. And for the cancers in the 5 cancer group, that was especially clear. So especially for these 5 cancers, we could see that a pretty high fraction of these NMF genes, if we looked at them by themselves, they would be able to split the tumors into 2 groups with different outcome. And when we looked at the prognostic genes from the based on that were by themselves able to separate the groups, we could see here that there was an enrichment, and that was enrichment for proteins that are important for histone modification, so histone methyltransferases, histone deacetylases, and the SWI/SNF complex, which is important for arranging nucleosomes. And if we look specifically at a couple of SWI/SNF genes, we could see that they themselves were very good at separating at least the top ones are very good at separating patients with better or worse prognosis. And you can see that for 3 different SWI/SNF genes for ACC and for LIHC. And we also wanted to take a look at these prognostic genes that were involved in the cell cycle and try to get a sense for the extent to which the correlations that we were observing in patient outcome might have to do with the cell cycle. So we learned about something called a meta-PCNA correction, where you look for all of the genes that are closely associated by gene expression with PCNA, which is a readout for how much cycling cells are doing, correct for that, and then look for and then we looked for whether we were still seeing significance. And we could see that there was a reduction, meaning some of this epifactor expression was capturing how much cycling there was going on within the cell, within the tumor. But here you can see that even after our correction, in many cases, many of these prognostic genes continue to be prognostic even after the correction. So some of the kind of prognostic capacity of these genes was through cell cycle, and some of it was independent. So then showing that we were able to divide these tumors into groups and that those groups had different outcome, we wanted to ask, can we develop some sort of algorithm that would allow us to predict if you get a new tumor, is that going to be in the better or the worst category? And so in order to do this, we used a neural network. We fed the neural network information about the counts for these epifactor genes and the age and sex of the patients. We gave the neural network 80% of the patients. And then the neural network had a hidden layer where it kind of took that data, did its analyses, told us, and then we said, okay, here's a new set of patients. When we give you this info, and in the first case, we told it like, this is high, this is low. It developed its model. Then we gave it the next 20% of patients. We gave this information and we said, which of these patients do you think is going to have better or worse outcome? And we looked to see, because we had the information on that patient, those patients, we looked to see, did they have better or worse outcome? And we found that when we made this neural net from these 5 cancer group and then fed the 20% that we had left out, actually the computer did a very good job, the neural net did a very good job of predicting patients with a high or low survival rate. And you can see the progression-free intervals were very different because we had that data, were very different when the neural net was based on the 5 cancer group. We had noticed that Kirk had seemingly somewhat pretty different gene expression pattern than the other 4. So we also made a neural net on just the other 4 without Kirk. And then actually we did even better. So giving the epifactor expression data and giving information on the outcome for the 80% and then feeding in just epifactor expression data, age and sex of the patient, the neural net could do a really good job of predicting whether that patient would have longer or shorter progression-free interval from a patient that we didn't give the outcome data to it. So that was very exciting. And we saw that this strong prognostic power of the top neural net genes. So if we gave each of these kind of top ML features here, we could see that each of these were individually quite informative. So just like for the machine learning, if we put in like individual genes and asked, the neural net gave us genes, and we could see that those by themselves could be pretty informative. That's shown here across the different pan-cancer types. So this shows once again what we were hoping, which was that we would be able to categorize information or come up with information that cross cancer types. And you can see, for instance, this NPAS2 was predictive across all 5 cancer types by itself, and this was identified by the neural net. And we wanted to see the genes that were identified by the neural net and the genes that were identified by the clustering, the NMF clustering, did they overlap with each other? And we saw that about a half of the top neural net network input features were also top NMF genes for some cancer type. So they weren't exactly the same, but they were similar. So then we wanted to ask, all of this clustering that we're doing, can we use it to predict outcome in independent data sets? So here we took the best independent data set that we had was also TCGA data because it had so much data, but we looked at the low purity tumors. And we asked the NMF epifactor expression clustering that we had done, if we apply that to low purity tumors for Kirk, would we be able to see a difference in the outcome? It didn't work for every tumor, but in the case of Kirk, we actually got really good predictive power. So it was sufficient to see a really good difference for progression-free interval and disease-specific survival. So this suggested to us that the clustering that we had an overfit, that it wasn't just specific for that if we took in new tumors, we'd be able to do a good job clustering them as well. So all of this had been done on adult cancer. We were also very interested in pediatric cancer. So pediatric cancer, we know, is also very difficult to treat. It can come from very different cell types, and so that's what we've chosen to study here. And we were particularly interested in pediatric tumors because they're known to have fewer mutations, so children just have had less time to develop mutations over the course of their life. And so it seemed like an opportunity to study a type of tumor that we expected a priori to perhaps be particularly epigenetically driven. And so for each of the 4 tumor types that we looked at, we could get clusters. And so this is for neuroblastoma and osteosarcoma. And again, we got 2 clusters. Again, 2 clusters were best based on epifactor expression. Here you can see for neuroblastoma and osteosarcoma, we got pretty good separation of the clusters based on just epifactor expression. And not for the other 2, so not for Wilms tumor or AML, but for neuroblastoma and osteosarcoma, we saw a big difference in outcome depending on whether the patient was in cluster 1 or cluster 2. So again, we're seeing that epifactor expression was sufficient to separate the tumors into 2 clusters, and those clusters had different outcome. And when we looked at the top NMF genes from the pediatric tumors, we saw that about a third of them were also top NMF genes in the adult tumors. So that suggests, again, we're seeing commonalities across different types of cells, across different types of tissue. The pediatric tumors and the adult tumors seem to have some commonalities, but the pediatric tumors seem to have some things that were kind of special for them and were not frequently observed in the adult tumors. Again, when we looked at the top NMF genes, similar to the adult tumors, we saw histone modifications and SWI/SNF as being some of the top enriched protein complexes. And again, when we looked at the specific genes that were indicative, looking at the entire genome, specific genes that correlated with the genes that were informative, again, we were picking up cell cycles. So we expect that some of our epifactor expression was picking up a cell cycle effect in these pediatric tumors as well. And again, we could make protein-protein interaction clusters, indicating that these epifactors that we're picking up likely interact with each other, and some of these indicated cell cycle also. So among the 24 epifactor genes that were prognostic, we identified some that individually could predict outcome. And here you see one for neuroblastoma, one for osteosarcoma. So this analysis allowed us once again to find individual genes that were sufficient to predict outcome for different types of tumors. So then kind of the final question that we wanted to ask had to do with how intertumor heterogeneity related to intercellular heterogeneity. So we used single cell sequencing to ask this epifactor expression pattern that we were seeing between the tumors. Can you see it in individual cells? And we could actually assign cells, here you see for LGG, for low-grade glioma, to having an epifactor expression pattern that was more associated with the poor outcome or more associated with the better outcome. There were cells that seemed to have both, but some cells seemed to go in one direction or the other, suggesting that this overall pattern of epifactor expression that we were seeing could be found on the cell-by-cell basis for individual cells within the tumor. And here we're showing it in the neuroblastoma from the pediatric patients as well, where we could see that there were some individual cells that had the epifactor better expression pattern, and some individual cells had an epifactor worse expression pattern. So just to conclude, for this group of 5 cancers, we found that 2 clusters that were generated from epifactor expression were different for progression-free interval, disease-specific survival, and overall survival. We found that more signature epifactor genes were prognostic for these 10 cancer, for these kind of 5 cancer types and a bigger 10 cancer type. Using machine learning, we developed a neural network for the 5 cancer group that combined was highly predictive of outcome. We found that among 4 pediatric tumors, we found clinically relevant clusters for neuroblastoma and osteosarcoma. We found that epigenetic similarities between the adult and the pediatric cancer types could transcend tissue similarity, which we thought was really important. And that single cell analysis showed that some individual cancer cells had the expression patterns of the poor or better outcome clusters. And overall, we're hopeful that epifactors might suggest treatments for specific cancer types, for instance, histone deacetylase inhibitors. And we did see kind of an expression pattern associated with histone deacetylases. So I just want to conclude by giving a big thank you, a huge thank you to all of our funding sources. Huge thank you again to Dr. Mithun Mitra, who's played a really instrumental role in all of this. Huge thank you to all of the members of my lab. And down here are undergraduate students who were working under Dr. Mitra's supervision, who did a fantastic job putting together all 3 of these projects. I'm hugely grateful to them and hugely grateful for the opportunity to be doing this exciting work. So thank you.
Hillary, thank you so very much. Wow, what an amazing amount of work. It is so impressive. And I was like really thinking about the stuff you talked about in like I think one of the middle experiments where you're really starting to dive into the tissue specificity of some of your epigenetic factors, and that the cell cycle ended up being like one of the big things that popped out of this, and that by looking at some of this, you actually do better than just tumor grading alone. And I wanted to have you comment a little bit more on that because tumor grading has been it's sort of the coin of the realm in a lot of cancers. And what this suggested to me is that through some of these analytical methods that you've been exploring, that you can do better than grading. And that may suggest that grading might be on its way out for other methods that are more precise. What are your thoughts on that?
I think I don't want to downplay the pathology. I think the pathologists do a fantastic job looking at tumors and giving you a sense for, um, how aggressive they are. I'm not trying to suggest that pathology is
Of
going to be
course, yeah.
supplanted at any point in the future because I think that the information that pathologists provide is extremely valuable. The thing that just this kind of brought home to me was that I think for me personally, I'd been kind of I mean, I come from a cell cycle lab, so I'd been very focused on the cell cycle. When I looked at a lot of these factors, all throughout this project, I'd been saying, that's a cell cycle protein, that's a cell cycle protein.
Mhm.
So I wasn't surprised to see cell cycle coming up. We went by the list of epifactors because that was like already previously established and documented list. So we went by the list that was there and we, didn't include or exclude based on our all preconceived notions. But I could see that a bunch of them were proteins that I had previously associated with the cell cycle. And that's not surprising. An important part of the cell cycle is replicating DNA and separating chromatin.
Yeah.
So it's not surprising that proteins that do stuff to DNA would be cell cycle associated. So I wasn't surprised that part of the signature that we were getting is taken out when we look at that meta-pCNA kind of analysis. Uh, and that some of those like prognostic proteins were kind of giving information that was concordant with the information you get from the cell cycle. We've done other studies that I didn't show you here where we're very interested in, like you said, quiescence and cancer dormancy. So we've done other studies where we're trying to identify like cancer cells that are proliferating, cancer cells that are not proliferating, what tumors having more proliferative cells is associated with bad outcome. So that's a project that's ongoing right now. And we could see that having more proliferating cells in general was associated with bad outcome, but even that was very tissue specific from our study so far. So I was actually surprised to see that in some cases, it wasn't actually true that having more proliferating cells was associated with bad outcome. So I think that, for me, I'm realizing our understanding of cell cycle and, cancer progression needs to be more nuanced. In some cases, like more cycling cells, like worse outcome, yes, that's true. But it's not always the case. And here it's showing me that, yes, cell cycle is important for bad outcome, but there are other layers on top of that too that, we're really just beginning to appreciate it. So I think it's not just having rapidly cycling cells, but probably also having chromatin conformations that facilitate tumor progression, either through DNA methylation or histone modifications or the placement of the nucleosomes, which really the data kept telling us was important. And that it's a combination of not just cycling more, which of course, tumors that cycle more, they're going to invade the tissue, that's going to be bad for the patient, but also having your chromatin arranged in a certain tumor promoting way is also bad for the patient.
Hillary Kohler from UCLA. Thank you so much. This is like a fantastic amount of work. This is really a great example of bringing kind of data analytics, methodologies to bear to try to understand something fundamental, keeping it real with the biology and not just kind of going all in on some of the story. And I think it's going to like be an amazing amount of work we're going to see out of your lab and from those of your colleagues as well. Thank you so much for sharing it with us today.
Thank you. It's my pleasure to be here.
Have a wonderful weekend, Hillary, and everyone who has been joining us.